There's a stretch of water beneath the sea ice of McMurdo Sound that sits at minus 1.9 degrees Celsius. Not near freezing. Below it, colder than the point at which the blood of almost every fish on Earth would begin to turn to slush. Lower a net into it and pull it back up, and you get fish. Alive, twisting, fighting the net. In the 1960s, a young American biologist named Arthur de Vries went down there to find out how that was possible, and he found the answer in their blood, a molecule that grabs hold of ice crystals and stops them growing. That discovery made it into the textbooks within a decade. What didn't make the textbooks, not for another 40 years, was the complication. Those fish are carrying ice inside their bodies right now, and nobody is entirely certain they can ever get it out again. Everything about the Southern Ocean bends back to that one strange fact that life down there didn't beat the cold so much as strike a bargain with it, and bargains have terms. So let's start with what de Vries actually found, because the mechanism is stranger than antifreeze makes it sound. The word itself is a bit misleading. The stuff in your car radiator works by brute force. You dump enough ethylene glycol into water, and you shift the freezing point of the whole solution downward, and it takes a lot of glycol to move it very far. What de Vries pulled out of Antarctic fish blood does something different. These are proteins, long repeating chains built mostly from two amino acids, alanine and threonine, with a sugar hooked onto the threonine. And they don't lower the freezing point of the fish's body fluids by any meaningful amount. What they do is find ice. Here's the thing about a growing ice crystal. It gets bigger by adding water molecules to its surface, layer by layer, and that surface has to stay reasonably flat for the process to be efficient. The antifreeze proteins bind directly onto that surface and stud it, like nails driven into a board. Ice can still grow, but now it can only grow in the gaps between the proteins, which means it has to bulge outward in tiny curved fronts, curved ice. Surfaces are thermodynamically expensive. The tighter the curve, the harder it is to add the next molecule. Push far enough and growth just stops. The result is a gap. The fish's blood will still melt at around minus 1 degree, but it won't freeze until you take it far below that, freezing and melting. And stop happening at the same temperature. Biologists call that gap thermal hysteresis. And outside of a handful of organisms, it basically doesn't exist in nature. So the fish isn't preventing ice from entering its body. Ice does get in, through the gills, through the gut, through the skin, and water that is already full of ice crystals. The proteins simply find each crystal and shut it down before it can grow large enough to do damage. That's an elegant solution. It's also a solution that raises an obvious question, and we'll come back to it, because the more interesting question for a long time was where a protein like that even comes from. Evolution is usually a matter of taking something that already exists and modifying it. New genes tend to be old genes with the serial numbers filed off, but a repeating chain of alanine and threonine with a sugar attached doesn't obviously resemble anything else. In a fish. In 1997, de Vries and his colleague Christina Cheng traced it, and the answer was so unlikely that it's now used as a teaching example in molecular evolution courses. The antifreeze gene came from the pancreas, specifically from trypsinogen, the precursor to a digestive enzyme that breaks down protein in the gut. A short 9-letter stretch of DNA inside that gene happened to code for the sequence threonine, alanine, alanine. At some point, that little fragment was copied, then copied again, and again, until it had been duplicated dozens of times into a long, stuttering repeat, and the gene it sat inside had lost almost everything that made it a digestive enzyme. What was left was a molecule with no obvious purpose, except that, by accident, it fit the surface of ice. A stutter in a digestion gene became the reason there are fish in Antarctica. Dating that event is harder, and this is where you should be careful with anything you read. The original estimate put it somewhere between 5 and 14 million years ago. Later work using different molecular clocks has pushed that window around considerably, in some cases much earlier. What we can say is that it happened while the Southern Ocean was cooling and before these fish spread across the continental shelf. The exact date is still moving. What makes it stranger still is that it happened twice. Cod in the Arctic, on the other side of the planet, carry an antifreeze glycoprotein that is nearly identical in structure. Same. Repeating unit, same function. It did not come from trypsinogen. It came from an entirely unrelated stretch of DNA. Now here's the part that gets glossed over in most retellings. It's tempting to describe all of this as a brilliant adaptation to the cold. A lot of specialists don't think that's what happened. The alternative reading is that losing hemoglobin was a genuine loss, a mutation that broke something, and that the Southern Ocean happened to be one of the very few environments on Earth where an animal could break that particular thing and still survive long enough to reproduce. Cold, oxygen-rich, low-competition water didn't reward the loss so much as it failed to punish it. The enlarged hearts and the wide vessels came afterwards as damage control. Under that reading, icefish aren't a triumph. They're an accident that the Southern Ocean was gentle enough to absorb. We can't currently settle that argument, and both camps are working with the same evidence, but it changes what you're looking at, and it raises an uncomfortable point about how narrow the window is that keeps these animals alive, which is going to matter later. If you drop a camera onto the seafloor down there, the second thing you notice, after the cold, is the size of things. Sea spiders are the standard example, almost everywhere in the world. These are tiny, a few millimeters, easy to miss, spindly little arthropods creeping over sponges. In Antarctic waters, the largest species reach leg spans of half a meter or more. There are isopods down there that dwarf woodlice anywhere else. Ribbon worms meters long, amphipods that would be crustacean specks in temperate water and are thumb-sized here. The leading explanation is called the oxygen temperature hypothesis. Cold water holds more dissolved oxygen. Cold animals run slower metabolisms and use less of it. Many of these creatures have no lungs and no efficient circulatory system. They rely on oxygen diffusing in through the body surface, and size is limited by how far it can diffuse before the interior runs short. Loosen that constraint and the ceiling rises. There's real support for it. A well-known study in the late 1990s found that across different bodies of water worldwide, the maximum size of amphipods tracked. Available oxygen too closely to be coincidence, but it isn't a closed case, and it's worth saying so. Later experiments putting giant sea spiders under warm or low oxygen conditions have produced mixed results, with the largest individuals not always struggling as much as the hypothesis predicts. Gigantism also isn't universal down there. Plenty of Antarctic groups are perfectly ordinary in size, and a few are smaller than their temperate relatives. Something about the combination of cold, oxygen isolation, and the sheer age of these communities allows giants. Exactly which factor is doing the heavy lifting is not settled. The other thing that changes is the clock. Everything runs slow. Antarctic marine invertebrates grow at rates that can be an order of magnitude below their temperate equivalents. Some sea urchins and scallops down there take decades to reach, a size their cousins elsewhere hit in a couple of years. Eggs are large and development is slow, and animals that would be short-lived anywhere else can live for a very long time. You'll sometimes see claims that certain Antarctic glass sponges are 10,000 years old, the oldest animals on Earth. Treat that carefully. Those figures came from extrapolating growth. Rates measured over short observation periods, and they've been challenged hard. Sponges of that type have also been observed growing far faster than the estimates allow, particularly after ice shelves collapse and light and food conditions change. Centuries is defensible. Millennia is a hypothesis that has not held up well. And then there's the absence. What isn't there is as important as what is. Look at almost any seafloor community in the world, and you find animals that crush shells for a living. Crabs, lobsters, rays, certain sharks, and bony fish. They've shaped marine life for well over 100 million years. And the reason so many seafloor animals are armored, buried, fast, or cemented down is that these predators exist. On the Antarctic shelf, they largely don't. Reptant decapods, the crabs and lobsters that walk, have been essentially absent for tens of millions of years, for reasons tied to the difficulty of handling magnesium in their body fluids at that temperature. Skates and shell-crushing sharks are scarce to absent. Remove that pressure and the community that develops looks wrong to a modern eye. It looks old. Dense fields of stalked crinoids, brittle stars and sponges carpeting open ground, soft-bodied, animals sitting exposed with no obvious defense, slow-moving grazers wandering across the surface. Researchers who've studied this have argued the Antarctic shelf resembles seafloor communities from the Paleozoic, hundreds of millions of years ago, before the crushers arrived and rewrote the rules. Not a relic in the literal sense, but a functional echo of a world the rest of the ocean lost. Two lineages, separated by the whole width of the Earth, both cornered by cold, both arriving at the same molecular answer from completely different starting material. But none of this explains the bigger question, which is why the Southern Ocean got so cold in the first place, and why it stayed that way while the rest of the planet moved on. For most of the past 100 million years, Antarctica was not what it is now. There were forests on it. There were dinosaurs and later beech trees and rivers and small marsupials. It sat over the pole, but it was connected. First to Australia, then to South America, and warm water. From the north, could still reach its coasts. Then the continents pulled apart. The gap between Antarctica and Australia opened. The Drake Passage between the Antarctic Peninsula and the tip of South America opened too. And once it did, there was nothing standing in the way of a current running all the way around. The planet at that latitude. No land to deflect it, no coastline to turn it back. What formed there is the Antarctic Circumpolar Current, and it is the largest ocean current on Earth by a wide margin. Depending on which measurement you use, It moves somewhere in the range of 140 to 170 million cubic meters of water every second, roughly 100 times the combined flow of every river on the planet. It runs clockwise, unbroken, forever, and it functions as a wall. Warm surface water from the Atlantic, Pacific, and Indian Oceans moves south, hits that current, and is turned. Around the point where it meets the polar water, there's a sharp transition called the Antarctic convergence, where surface temperature can drop by 2 or 3 degrees over a very short distance. That's not a gradual gradient. For an animal adapted to one side of it, the other side is effectively another planet. Now, the older textbook version of this story said, the current is what froze Antarctica. Thermal isolation, full stop. That version is no longer safe to state as fact. Climate modeling over the past 2 decades has made a strong case that falling atmospheric carbon dioxide was the primary driver of Antarctic glaciation, but the current amplified and locked in a change that was already underway. The dates for when the Drake Passage actually opened wide enough to matter are also disputed, with published estimates spread across tens of millions of years. What isn't disputed is the outcome. By something like 34 million years, Hago, ice sheets were forming, and the Southern Ocean was on its way to becoming a sealed room. And sealed rooms do interesting things to whatever is left inside them. Most of the original Antarctic fish fauna died. Cold that's severe, arriving over that timescale, cleared the shelf of nearly everything that had been living. There. What survived was a group called the notothenioids. Unremarkable, bottom-dwelling fish with no swim bladder, the kind of animal that, in most oceans, stays small and stays down and doesn't amount to much. With the competition gone and the antifreeze problem solved, they took the entire ocean. Today, there are somewhere around 120 described notothenioid species. On the Antarctic shelf, they make up roughly 3/4 of the fish species present, and well over 90% of the fish biomass. Some still live on the bottom, as their ancestors did. Others became mid-water hunters or krill specialists, and some evolved lipid deposits and thinned-out bone that let them hover in open water without a swim bladder, a trick most fish lineages never manage. It's the marine equivalent of what finches did on the Galapagos, except across a whole ocean. Which brings us to the strangest thing that came out of it. In 1928, a Norwegian biologist named Ditlev Rustad, working off Bouvet Island, hauled up a fish he couldn't place. It was pale, almost translucent, and when he cut it, the blood that came out was not red. It was clear, faintly milky, like watered-down cream. The local whalers apparently already knew the animal and had a name for it. Rustad wrote it up, drew it, and it sat there as a curiosity for a quarter of a century. It took until 1954 for another Norwegian, Johan Ruud, to publish the finding that should have made headlines everywhere. This fish has no hemoglobin. None. It has essentially no red blood cells. It is, as far as we know, the only vertebrate on Earth that carries oxygen around its body, without the molecule that every other vertebrate depends on to do the job. There are 16 species in that family now. We call them icefish. Hemoglobin is not a small thing to lose. It's the reason your blood carries something like 70 times more oxygen than plain water would. Take it away, and the blood of an icefish carries roughly 1/10 of what its red-blooded cousins manage. On paper, this is a lethal condition. The reason it isn't lethal comes down to a stack of compensations, plus a very large helping of luck about where they live. Cold water holds much. More dissolved oxygen than warm water, so there's more available to begin with. Icefish are sluggish, with very low metabolic rates, so they need less. They've compensated on the plumbing side, too. Their hearts are enormously enlarged relative to body size. Their blood vessels are wider. They carry several times more blood by volume than similar fish, and they pump it around far faster at much lower pressure. Their skin is scaleless and unusually well supplied with blood vessels, and there's good evidence they absorb a meaningful amount of oxygen straight through it. Some species have gone further and lost myoglobin from the heart muscle as well. In 2010, a submersible survey in the Palmer Deep off the Antarctic Peninsula found a population of king crabs on the slope. Thousands of them. And the immediate question was whether warming water had allowed a crusher back through a door that had been shut for 30 million years. And what a community. With no defenses would do if it opened all the way. That interpretation has been contested since. Other researchers have argued there's no solid evidence these crabs are new arrivals rather than a population that has always been there unnoticed on deeper slopes. The disagreement is still live and it matters, because the two readings point at very different futures. Which brings us to the discovery that reframed a lot of this, and it's recent. In February 2021, a research vessel in the Weddell Sea was towing a camera system over the Filchner Trough, several hundred meters down, in an area nobody expected much from. The plan was routine seafloor mapping. And what came up on the monitors was a nest, a circular depression in the mud about the width of a dinner plate, with a single icefish sitting in it guarding eggs. Then another. The camera kept moving and the nests kept coming, and they didn't stop. They ran for hours. They ran for kilometers. The team eventually surveyed an area of around 240 square kilometers, and extrapolating from the density they measured, estimated something in the region of 60 million active nests. It is the largest fish breeding colony ever found anywhere on Earth, by an enormous margin, and until that afternoon, nobody had any idea it existed. There are things about it we can measure. The nests are spaced with striking regularity. Most held a guarding adult and something like a couple of thousand eggs. The colony's boundaries appear to line up with a body of slightly warmer deep water, flowing across that part of the trough. Tracking data from Weddell seals in the region shows them diving repeatedly over the colony, which suggests a food source of that size doesn't go unnoticed. And there are things we simply don't know. How long it's been there, whether it's one of many or genuinely unique, what the fish do the rest of the year, how a colony that size coordinates spacing across a landscape none of its members can see the whole of, whether the biomass of that many adults and eggs is a significant part of the Weddell Sea's carbon cycle. The paper raised those questions. It did not answer them, and it was honest about that. 60 million nests found by accident. In an ocean we've mapped less thoroughly than the surface of Mars. So, back to the ice. That question from the beginning about whether these fish can ever get the ice out of their bodies. In 2006, Christina Chang and her colleagues went looking for where the crystals end up and found them concentrated in the spleen, apparently swept up and stored there by immune cells. That looked like a reasonable answer. Ice gets in, gets bound, gets collected, gets held somewhere it can't do harm, and presumably melts out when the water warms a little in summer, except it doesn't, or at least not reliably. In 2014, the same research group published something genuinely peculiar. The antifreeze proteins that coat those internal ice crystals don't just stop them growing, they also make them resistant to melting. The ice inside these fish stays solid at temperatures above the point where it should have turned back into water. A state physicists call superheating, which is rare enough in a laboratory and was not expected inside an animal. Summer in McMurdo Sound doesn't get warm enough to overcome it, which means that for at least some of these fish, some of that ice may never leave. It accumulates. They spend their whole lives carrying it, and they may die carrying it. And what that costs them over a lifetime is not something anyone has been able to measure. That's the bargain. The molecule that lets them live in water below freezing is the same molecule that won't let go of what it catches. And that's worth sitting with, because the fish that made this deal have almost no room left to maneuver. They are among the most temperature-restricted animals ever measured. Some Antarctic species lose the ability to stay upright when the water reaches 5 or 6 degrees Celsius, a temperature you'd describe as cold, and a temperature that would kill them. Many have lost the standard cellular heat shock response that most animals use to cope with thermal stress. They didn't need it. For 30 million years, nothing down there got warm. The Antarctic Peninsula and parts of the Southern Ocean are now among the faster warming regions on the planet. Krill, the animal nearly everything else in that ecosystem eats, directly or indirectly, appear to have shifted their distribution southward in the Atlantic sector over recent decades, and there's an active debate about what's happening to their overall numbers. Sea ice extent has become erratic in ways that don't fit earlier expectations. The animals of the Southern Ocean exist because a wall of moving water closed a door and gave them 30 million years alone. Nothing else on the planet had that. It's why the sea spiders are enormous, why the seafloor looks like a fossil, why there is a fish with clear blood, why 60 million nests could sit undiscovered on a mud plain. It's also why they have nowhere to go. Every one of these solutions is a specialization to a set of conditions that has held steady since before our ancestors came down from the trees, and specialization is only ever a bet that tomorrow resembles today. The door is being tested. We don't know yet how firmly it's shut.